Hello everyone. Today I am going to explain basic introduction to DNA and its components along with its type, structure, properties, function and small amount of information about drug DNA interaction. So as you people can see on the screen, the DNA stands for deoxyribonucleic acid, is a molecule of great biological significance. The total DNA content of a cell is termed as genome. The genome is unique to an organism and is the information bank governing all the life processes of the organism. DNA being the form in which the information is stored, structures of DNA called genes have the extremely important function of coding for proteins. हमारा जो डीएनए होता है वो हमारा यूनिट ऑफ हेरिडेटरी, जिसको हम जीन्स कहते हैं, उसको कंटेन करता है और जनरेशन टू जनरेशन ये हमारा डीएनए ही है जो पास होता है और हमारे जेनेटिक इंफॉर्मेशन को कैरी करता है. Deoxyribonucleic acid is the hereditary material found in the humans and all living organisms. Of course, it is found in humans and all living organisms. But in viruses, there is either DNA or either RNA, but not both. Okay? Because the first genetic material which was found was ribonucleic acid, that was RNA. The discovery of RNA is the prime genetic molecule carrying all the information within chromosomes immediately had its attention focused on its structure. As we know that there were several experiments carried out for knowing what is our hereditary material. The first experiment was carried out by Frederick Griffith in 1928, and it was known as transforming principle experiment, in which he used pathogenic and non-pathogenic, that is also called virulent or non-virulent strains of bacteria, and he used Streptococcus pneumoniae in that to find and he found that there is something that is being transferred from heat killed S strain to non-virulent strain, and the mouse, which was the experimental organism, was died. Then there was an experiment of Hershey and Chase using transduction experiment using a bacteriophage, and ultimately we got to know that DNA is the only hereditary material that is being transferred from one generation to the other. Now let's see the components of DNA. DNA is made up of nucleotides. Each nucleotide has three components: a backbone made up of a sugar and a phosphate group and a nitrogen containing base attached to it. There are four nucleotides that is given here: adenine, guanine, cytosine and thymine. And in RNA we have uracil. We have uracil in place of thymine. We have uracil in place of thymine in RNA, whereas we have thymine in case of DNA. Okay? There are two kinds of bases. One is first is purine and the other is pyrimidine. Purine base contain adenine and guanine, whereas pyrimidine base contain cytosine, thymine. Okay? So, and the uracil is also present in pyrimidine base, present as pyrimidine base only, but it is present in RNA. Okay? Point to be noted, uracil is present in RNA, not DNA. It is an extra information that I am giving to you people. Okay, now let's see about deoxyribose sugar. Deoxyribose sugar of the DNA backbone has 5 carbons and 3 oxygen. Okay, as you people can see in the diagram given beside, there are 5 carbons and 3 oxygens. Okay, and this sugar forms sugar phosphate backbone of DNA, and that is made by phosphodiester bond. DNA backbone is a polymer with an alternating sugar and phosphate sequence, also known as phosphodiester bond. Here we can see that adenine always binds to thymine. Adenine always binds to thymine via two hydrogen bonds, whereas guanine binds via 3 hydrogen bond to cytosine. Okay, now let's see types of DNA. aDNA, bDNA, zDNA. aDNA and bDNA are right-handed helix, that is their orientation is via right side, whereas zDNA is left-handed helix. Okay, this is the very common differentiation between these types of DNAs. Now let's study the structure of DNA. As we know, the structure of DNA was found by Watson and Crick in their experiment in 1953. Okay, and the experimental proof of Watson and Crick experiment was given by Messelson and Stahl by their semi-conservative mode of DNA replication. So DNA replicates, you all can note that DNA replication is carried out in a semi-conservative manner, I'll explain you people what does semi-conservative means in further. Okay, DNA is made up of two helical chains that intertwine with each other to double helix. The most widely accepted structure of DNA is right-handed helix. Obviously it is right-handed helix, and most widely accepted DNA structure is bDNA. What it is bDNA, as it is written also further that it is bDNA, and the whole also known as bDNA, which is 1.9 nanometer in diameter, and the total length of DNA in a human cell is 2 meters, about 2 meter. We can call it 2.2 meter, but generally we say 2 meter. Okay, it contains two polynucleotide strands that binds around each other. The backbone of each consists of alternating deoxyribose and phosphate groups. We can call it sugar phosphate backbone. What we can call is sugar phosphate backbone. Sugar phosphate backbone, and as I have told you people that it is made up of phosphodiester bond. What it is made up of? Phosphodiester bond. The phosphate group bonded to the 5' carbon atom of one deoxyribose is covalently bonded to the 3' while the other runs 3' to 5'. Here you all people can note that the DNA replication is carried continuously in the orientation of 5' to 3', whereas in 3' to 5' it is carried out in a discontinuous fashion that you'll study further. The purines and pyrimidine attached to each deoxyribose projects in toward the axis of the helix. Okay, that is projected inwards. This sentence means that the bases are projected inwards towards the helix. That it means that in this manner. Okay, and as I have told you people that adenine binds to thymine in via 2 hydrogen bonds, whereas guanine binds to cytosine via 3 hydrogen bonds. Each base forms hydrogen bonds with the one directly opposite to it, forming base pairs, and this is known as base pairs. Let's come to the properties of DNA. DNA is hereditary material found in almost all prokaryotes and eukaryotes, obviously, but in viruses, it is either DNA or RNA, not both. This point should be noted. It is composed of two helical strands and are wound around same axis. DNA is polar in nature because of its highly charged phosphate sugar backbone. Obviously, phosphate is present, so it is negatively charged. And DNA bases are capable of absorbing UV light at 260 nanometers. And this, you can know that DNA absorbs 260 nanometer by using a technique known as spectrophotometry. Okay, spectrophotometry. DNA bases are capable of absorbing 260 nanometer. You people know that, and we check absorbance in spectrophotometer. That is based on Beers and Lambert's law that you'll study further in your studies. DNA denatures on being subjected to high temperature. What does it means? That is, the hydrogen bonds get broken, okay, and the two strands of DNA get separated, and this is the property that helps in many experiments, replication of DNA, PCR technique and all. On cooling, the two separated strands join each other. This process is called renaturation. Okay. Function of DNA. Transcription. What is transcription? Transcription is a process by which DNA is being replicated, or DNA is being converted to RNA and especially to mRNA that codes for a protein, and that is based on genetic code. What is written here is information is retrieved from the DNA by ribonucleic acid, RNA, and utilized to synthesize proteins in the body. As I have told you people, proteins are involved in all body processes and play many roles. Example as hormones, enzymes, carriers, structural protein receptors, regulators, etc. Replication. DNA is responsible for its own regeneration, that is, DNA self replicates. Here I want to show you people via flow chart that DNA convert to RNA via transcription, and DNA convert to DNA via replication, and then RNA convert to protein via translation. Okay, so DNA convert to DNA via replication. DNA convert, sorry, 1 minute, it will be DNA convert to RNA via transcription, RNA convert to protein via translation, and DNA self-replicates via process of replication, and that is carried out using DNA polymerase enzyme, So that's it. It is enough for introduction of DNA. Thank you.